introduction. And yeah, so this talk is about using deep generative models to design polyadenylation signals. And you know, even if you're not super interested in the specific mechanism, polyadenylation, I think this might be, one potential, good recipe for doing molecular sequence design for some other genomic regulatory control elements that you want to engineer. And I'd like to begin by just acknowledging the great collaborators I've been working with in Seelig Lab. So, for example, my PI, Professor Georg Seelig, and also in particular, Dr. Nicholas Bogard, who's a postdoc in the lab and who's the co-author on some of these papers that I'm going to talk about. And he's also the person who's been doing all of the wet lab experiments that I'm going to show you. Beyond that, I'd like to thank NIH for funding this research. So here's an overview of the talk today. We're going to start by quickly going through sort of a background on what alternative polyadenylation is and why it's important. And then we're going to sort of define, the problem of molecular sequence design and, what we want to get from that. Then we're going to go through the data set that's used in this study. And essentially, it's a large reporter assay measuring millions of randomized APA events. We take this large data set and we train a sequence-based convolutional neural network called APARENT. And this predictor is used to sort of predict alternative polyadenylation. Once we've sort of gone through the data and the predictor model, we're going to sort of get to the heart of this talk, which is about designing polyadenylation signals, new and improved ones. And we're going to use a technique called activation maximization, which is essentially gradient descent, to design new polyA signals. And we're going to start with just the basic principle of activation maximization, which we used in one of the papers to design polyA signals. And then we're going to sort of discuss an extension of activation maximization into generative neural networks. So let's jump into the background. So polyadenylation, without the word alternative before it, is just a mechanism that happens at the 3' end of all human mRNA molecules, where a sequence regulatory code called the polyA signal, shown here as this green box in the 3' UTR, this signal essentially attracts the polyA machinery, which is a large protein complex that comes in and cleaves the RNA molecule at that position and appends a long string of A's. And the polyA signal, which is composed of this cis regulatory code, essentially regulates the strength of attracting this polyA machinery by incorporating various RBP binding sites, as well as having a particular RNA secondary structure, perhaps, to regulate its strength. Now, alternative polyadenylation is the phenomenon where you have multiple competing polyA signals within the same RNA molecule. And in fact, more than 50% of human genes have more than one polyA signals within them. And the relative abundance between these, the isoforms that result from these different polyA signals depends highly on this internal strength of the polyA signal. It's this regulatory code. And alternative polyadenylation, in general, is an important mechanism to study because numerous genetic disorders have been linked to it, such as alpha-beta thalassemia and thrombophilia, to mention a few. So the sort of fundamental question we ask is, given the primary sequence of these polyA signals, can we predict cleavage and polyadenylation? And rational sequence design is important for engineering purposes because it would allow us to design very specific regulatory control elements that we could incorporate in synthetic gene circuits. So for example, with sequence design, we can engineer poly-A signals, for example, for strong polyadenylation, which results in strong, efficient termination of RNA molecules. Or we could use it to engineer target isoforms with differential stability in our genetic circuits. In terms of splicing, we could use it to engineer differentially used coding isoforms depending on cell type. And in terms of transcription, rational sequence design could be used as a toolbox to engineer stronger differential expression. And all of these control elements would be really important to design for our synthetic gene circuits, because we could then use them to improve experimental assays or components within metabolic engineering, or to improve drug delivery vectors and systems in smart therapeutics. So today we're going to sort of look at the specific story of designing poly-A signals. But in these couple of different papers that our lab has put out, we do look at engineering other types of regulatory mechanisms as well, for example, splicing and transcription. And for anyone interested, I urge you to look at those papers for studying those mechanisms. So in a nutshell, given our pre-trained differentiable fitness predictor, this model that we're going to train on our data, the goal is to use that predictor to design new and improved sequences with improved biological function, which in this case is alternative polyadenylation. And we're going to use gradient descent essentially as the workhorse for designing sequences by doing backpropagation through this differentiable fitness predictor, and in a sense optimizing the input pattern and using that as a means of designing sequences. And this technique is commonly referred to as activation maximization. So now let's look at the data set that's used here. What it is, it's a massively parallel reporter assay of random APA variants. And this was a library that was constructed by Dr. Nick Bogard in the lab. Essentially, at a high level, we make this large plasmid library where we introduce degenerate oligonucleotide regions near poly-A signals in the 3' UTR of these plasmids. So you see sort of a zoom in on the 3' UTR of one of these plasmids, where you see two competing poly-A signals indicated with the green and the red small boxes. And then you see these N20 boxes, which is one example of a type of degenerate regions we can insert near the poly-A signal if we want to study the effect of varying the regions surrounding the poly-A signal. So now when we have this large plasmid reported library with targeted variation near the poly-A signal, also I should mention that for this library, we don't actually vary the distal signal. We only vary the proximal poly-A signal here. It turns out that we can learn a good general model of polyadenylation, for predicting poly-A in general, even if we just learn what proximal poly-A regulation looks like, because it's very it's regulated in the same fashion in the distal site. So we take this plasmid library and we put it through we express it and we transfect it and express it in a HEC cell line. These plasmid reporters are going to get expressed, and the resulting RNA is going to get polyadenylated. We can then extract the resultant RNA isoforms and sequence them, allowing us to estimate for every plasmid reporter, we can estimate the resultant polyadenylation isoform ratio. And now when we have this large labeled data set, we can essentially apply a machine learning framework to learn to predict the resultant isoform abundance given the sequence of a poly-A signal. So Nick constructed many different libraries, plasmid reporter libraries, with different sort of sites where he, relative to the poly-A signal, where he targeted, inserted degenerate regions in order to get a lot of variation. All in all, he constructed 12 different APA reporter libraries with a total of more than 3.5 million random APA variants. And again, using next generation sequencing for every one of those APA variant sequences, we could measure the resultant isoform expression. And in fact, even we could measure the cleavage distribution, the percent cleavage at nucleotide resolution for every sequence. So with this data set, we trained a sequence-based convolutional neural network to predict the cleavage and isoform distributions given the proximal sequence as input.
So there's a question for you, Johannes.
Yes.
So Matthew asks, did you try running your degenerate APA signal libraries in cell lines other than HEC293?
We did not. It's a good question. We did not run. That's something that we're looking at doing, and that we're sort of working on. We're interested in running our plasmid reporter library in other cell lines than these immortalized cell lines.
So remember from your paper that you did run your 5' alternative 5' splicing signals in other cell lines? Exactly.
So a similar plasmid reporter library of splicing events was run in different immortalized cell lines like HEC, MCF7, CHO, and HeLa. And it's great for validation purposes, but if we want to study differential APA between cell types, you really want to look at more like primary cells versus heart tissue cells. I think it's still a topic of research in order to actually be able to efficiently express these types of reporter assays in more complicated cell types.
Okay.
Does that answer your question?
Yes.
Great. Okay, so yes, let's get to this predictor model. And so a brief primer of what convolutional neural networks are. First of all, we're going to represent our input sequence to this neural network as a matrix, a one-hot coded matrix, where essentially the columns encode different nucleotide positions and the rows encode different nucleotide identities. This one-hot coded sequence representation is fed into the neural network, which consists of many layers of weighted units and is trained by backpropagation in order to learn how to learn a good representation of the input signal such that it can perform some prediction. And what's kind of special about convolutional neural networks is that the first layers are these convolutional layers consisting of these trainable filters. Now, the very first convolutional layer, when executed on a DNA sequence, corresponds really well to essentially motif detectors. They learn to detect important motifs. And the later layers in this neural network, they're going to learn to combine these motifs in order to make some nonlinear prediction in the end. So our particular network architecture for this predictor that we call APARENT is a convolutional neural network with two convolutional layers, followed by a dense or fully connected layer above that. And on the top, we have two different outputs. We have a sigmoid output that predicts the isoform abundance given this proximal poly-A signal's input and some fixed distal poly-A signal. And it also predicts at a nucleotide resolution the cleavage distribution for this sequence. So where cleavage is going to happen across this sequence. And so we trained this neural network on 9 of 12 of these Munigene reporter libraries and held out 3 of these libraries for sort of independent validation. And we trained the network essentially by minimizing the KL divergence between the predicted and target isoform abundances, as well as the predicted and target cleavage distributions. So after we train the network, we can evaluate it. The first test, is to have your held out test set of other plasmid reporters that you didn't train on. As we can see, the predicted versus observed isoform proportions correlate really well. But we've trained this neural network purely on synthetic randomized APA sequence variants. One important check is to see, okay, does this network generalize to human poly-A signals, like human native poly-A signals? To that end, we took roughly 1,000 native human poly-A signals from the genome and put them in our plasmid reporter assay in order to measure their isoform proportion. And as you can see, the predicted isoform proportions of this APARENT predictor correlates really well with the observed usages without, retraining or fine-tuning the model, which I think, oh yes.
I have a question here. So how did you select the 1,000 events or signals? Like what was the selection criteria?
So the selection criteria was that, so we sort of got these human poly-A signals for free. Really, what we wanted to do, we wanted to show that APARENT could predict clinically relevant mutations and variants. Essentially from ClinVar and HGMD and GWAS, we wanted to show that it could predict variants. And so to that end, we had to, so we did saturation mutagenesis of these 3' UTRs that harbored these variants. And as a side product of that, of course, we assayed the wild type sequences as well, which gives us this large set of native human poly-A signals that, these wild type sequences, they're reported in ClinVar as having mutations near poly-A signals. So it's sort of clinically relevant poly-A signals in a sense.
Okay, there's another question from Yusuf. What happens if the random sequences create new polyadenylation sites? It seems the sites themselves are predefined in the network architecture.
Yes, that's a good point, and I think it's all a matter of how we define a poly-A signal. A poly-A signal is kind of loosely defined, right? If you have multiple, so essentially a poly-A signal is mainly governed by its central hexamer element, which is a hexamer element that sort of defines the poly-A signal. And if you have multiple of those, you're going to get sort of kind of spurious polyadenylation and cleavage. Like it's not going to be just one well-defined cleavage site within this 1,000 nucleotide region in the UTR or whatever, but you're rather going to get these multimodal cut distributions. And so the APARENT network, the isoform output itself treats these spurious cleavage events that are upstream or too far downstream from this central hexamer that we aligned on, treats that cleavage as competing cleavage, like it counts that as the distal isoform, which means that the network will learn that these other hexamers that are close by, they're treated as repressive elements. However, this cleavage output actually, it learns to predict those spurious cleavage sites, which means that it does learn to incorporate these close by competing poly-A signals. So we can take that information into account as well. Did that answer the question?
Yeah.
Perfect.
Okay.
Okay. So unfortunately, I don't have too much more time to talk about this predictor neural network, APARENT. But in our paper, we showed that it is very accurate at predicting human APA variants, at least in the 3' UTR. That's what we tested it on. And for people who are interested in variant prediction, I urge you to check out our interactive web tool on apa.cs.washington.edu, where we allow users essentially to look at any position in the human genome and use this neural network to perform in silico saturation mutagenesis to study the effect of any potential variant in the genome with regard to nearby poly-A signals. So now we're going to sort of get into the heart of this talk, which is about using our trained polyadenylation predictor to design new and improved polyadenylation signals. And the method is activation maximization. Of course, molecular sequence design has been around long before me, and there's some really cool related work that I think is worth going through quickly. Many of these methods rely on this assumption that you have a pre-trained fitness predictor, or a predictor oracle, as it's sometimes called, that allows you to score an input sequence based on your, with respect to this biological function that you're trying to optimize. And so the general paradigm is that you start with some initial sequence design, and you then computationally evaluate its fitness using this predictor, and then make informed local changes to the sequence in order to iteratively optimize it. So classically, there's been a lot of work on developing discrete search algorithms in sequence design space, such as evolutionary algorithms that performs in silico mutations randomly in your initial sequence design in order to accumulate positive mutations using your predictor as a proxy for knowing which mutations are positive. Then there's more intricate heuristics like genetic algorithms and simulated annealing that allows you to overcome some of these local minima in the fitness cost landscape during optimization. Lately, there's also been a lot of work in adapting these distribution capturing deep generative models, such as generative adversarial networks and variational autoencoders, to molecular sequence design. The general scheme here is that you take your initial set of sequences and you train, for example, a GAN on that data, allowing you to hopefully capture the manifold along which functional sequences reside. And you can then use this trained GAN to sample or hallucinate new, hopefully functional sequences that are on this manifold. And there's been some really cool methods developed sort of as a generalization of deep generative models, such as feedback GAN or conditioning by adaptive sampling, that combines deep generative models with in silico directed evolution to iteratively retrain and refine these deep generative models such that they get conditioned towards high fitness basins in the fitness cost landscape. Here we're going to take a slightly different approach to these other categories of molecular sequence design. In a sense, what we're going to do is something quite similar to the discrete search algorithms, in the sense that we're going to try to directly optimize the fitness predictor. However, we're going to assume that the fitness predictor is differentiable, such that we can perform backpropagation through the predictor in order to optimize the input pattern to that predictor. And that input pattern happens to be the sequence that we're trying to design. And this technique is referred to as activation maximization. So essentially, the problem here now is that we start with some random initial sequence, again, which is typically represented as these one-hot coded sequence patterns. And the goal is to iteratively, using gradient descent, maximize this one-hot coded pattern such that we maximize the predicted fitness score. And the predictor in this case is our polyadenylation predictor, APARENT. Of course, a one-hot coded pattern is discrete and not amenable to gradient descent in its basic form. So the fundamental idea is to try to optimize a continuous sequence relaxation with gradient descent instead. So here we're going to initialize a matrix with random nucleotide logits. So this is a matrix of the same shape as a one-hot coded matrix, but it has real values in its different matrix entries. And these entries correspond to the log probability of a nucleotide at a particular position. We then need to transform these nucleotide logits into some continuous sequence representation. Killeron in 2017 used a softmax layer in order to transform these real valued scores into a matrix of nucleotide probabilities, or a sequence PWM. This sequence PWM is then fed straight into the fitness predictor, allowing us to evaluate this fitness objective that we're trying to optimize. Now, because this whole pipeline is differentiable, it allows us to backpropagate the gradient of the fitness objective all the way down to the nucleotide logits, allowing us to take a small step in the direction of the fitness gradient to update our nucleotide logits. Now, a potentially big problem here is that our fitness predictor has only ever been trained on proper, discrete, one-hot coded sequence patterns. It's never seen these continuous sequence relaxations before, which means that it may in fact be arbitrarily bad at accurately predicting those continuous representations. In particular, when the softmax PWM has high entropy, that problem is more apparent. So what we did in one of our papers, the cell paper, is that we modified this design method, and we call this modified algorithm SeqProp, or stochastic sequence propagation. We introduce a discrete sampling layer in between this softmax representation and the fitness predictor, where we draw and construct proper discrete one-hot coded patterns from this softmax representation, effectively removing any potential softmax pathologies, because we're now feeding these one-hot coded patterns to the predictor. Of course, the discrete sampler, by its very nature, is not directly differentiable. However, what we do is we use a straight-through gradient estimator, where we essentially pretend that the gradient of the sampling function is the gradient of the softmax distribution. This allows us to propagate a gradient estimate down to our nucleotide logits and keep using gradient descent to optimize the sequence pattern. However, a separate problem that we noticed is actually apparent for both the original softmax method and the sampled SeqProp method is an artifact I call logit drift. And what this is, in a nutshell, is that during the course of optimization, these nucleotide logit values can grow overly large and get skewed with respect to one another, which can severely halt convergence when we're designing sequences or doing this optimization. So in a recent preprint that we just posted, we introduce this kind of subtle architectural change where we add a normalization layer in between these nucleotide logits and the softmax layer, allowing us to normalize and properly scale these logits, sort of overcoming this issue. And we're going to look at what this normalization scheme does with sort of an illustrative example. So you can imagine that, let's say for the first 10,000 iterations of this activation maximization procedure, 10,000 iterations of gradient descent, let's say that for all those iterations, there's one particular motif that's always been sort of promoted by the predictor. Like the predictor always says that, you should try to make this motif at this particular location because it's predicted to result in high fitness. During the course of optimization, the nucleotide logits will surely grow very large and very skewed towards favoring that motif, right? But then, you know, this is nonlinear optimization we're doing here, right? Which means that all of a sudden, maybe after 10,000 iterations, all of a sudden the predictor might now see another motif popping up somewhere else in the sequence, which has a nonlinear effect of saying that, okay, now some other motif needs to occur at this position, which means that the gradient descent machinery now has to try to reverse these very skewed parameters in order to let another motif enter. And this can, you know, take a very long time to do. So to remove this type of artifact, we introduced this normalization scheme where we normalize logits across position such that they have 0 mean and unit variance in expectation across position. Now you can, with very few gradient updates, you're always able to flip, you know, every nucleotide distribution very fast. Of course, nucleotide logits with 0 mean and unit variance has limited expressiveness when used as parameters to probability distributions. So to balance or trade that off, we introduce a global offset and scaling parameter. So these are two additional parameters that we actually train with gradient descent as well. And what the scaling parameter does is it's going to provide a multiplier in order to scale up the logits globally across all positions, which effectively means that the scaling parameter proportionally controls the entropy of the entire sequence PWM. So this sort of trades off, you know, and allows the optimization to maintain proportionally scaled logits across all positions during the course of optimization. And this turns out to result in a highly, a very fast design method. So in this first sort of benchmark test, we used our predictor APARENT to try to design maximally strong poly-A signals. And what I'm showing here is a sequence PWM that's been optimized using this normalized SeqProp method, starting from a random sequence. And this is the result after just 200 iterations of gradient descent. So already you can see many important motifs popping up, like the CFI binding motifs, the CPSF binding motifs, CSTF binding sites, cut sites, etc. And if we compare it, the optimization to the previous methods, the softmax method, which is the blue curve, or the non-normalized SeqProp method, which is the green curve, you see that this normalized SeqProp, which is the red curve, has a lot, converges to a much better fitness cost. So in this graph, we're plotting the negative of the fitness score. So lower is better. You can see that, I mean, so already after 200 iterations, it reaches a very good, it converges to a very good minimum, whereas these competing methods take, you know, on the order of hundreds more iterations to reach the same optimum. For this for this task, we wanted to use APARENT to design poly-A signals that cleaved according to a very specific target isoform proportions. Essentially, we wanted to show that we could design signals with specific isoform abundances. And so on the right side here, you're seeing some of these example sequences that were designed with this method. And as validation, we took these generated sequences and we synthesized them on these plasmid reporters and expressed them in our hex cell line and used sequencing to, again, read out the resulting isoform proportions. And so we collected, I think, more than 1,200 designed sequences for a range of different target isoform proportions. And as you can kind of see in this violin plot, our engineered target expressions correlated really well with the observed measured isoform proportions. You can sort of see this ladder, essentially, of these engineered expressions. And I think also what's really cool is that, you know, poly-A signals engineered for just maximally strong polyadylation turned out to be stronger than native poly-A signals. For this task, we wanted to see if we could engineer poly-A signals that cleaved at very specific target positions. So in these 3 example sequences that you see that the central hexamer signal, so the core polyadenylation signal, that position shown on the far left in the sequence, that's kept fixed all the time. But then we essentially vary the target cut position, and then we try to design sequences which maximize the predicted cleavage at that location. And we did that for longer and longer cut positions. And so I think it's really cool that you know, we could do this. So again, we synthesize these in a hexagon line, and what you see are these average cut profiles for various engineered target cut positions. So we could in fact design sequences with even, you know, cleaving that cleaved, you know, up to 45 nucleotides downstream of the hexamer. For these, you know, shorter target cut positions, we could see that the network learned to use these, you know, RNA binding protein motifs to direct cleavage. What I think is also really cool is that when we tried to, you know, design sequences that maximize cleavage very far away from the central hexamer, the network has learned that it must create these stable secondary structures like hairpins that, as you can see in the 3rd sequence, that essentially brings together the cut site and the CSE hexamer in order to initiate efficient cleavage.
Johannes.
Yes.
So can you talk a little bit about the biological plausibility of the sequence that are sequences that are being generated? Because I mean, you could have multiple evolutionary constraints that limit the sequences that you will see in a genome.
Right.
So
for
do
sure.
you address that somewhere? Do you evaluate?
I don't think we never really looked through that lens. I mean, I think it's a really, really great question, and that's important for some purposes, right? That the sequences are biologically plausible, and you know, but for this study, we sort of didn't look at that. We didn't look at that. Essentially, we just, you know, given our you know, synthetic data set, we got this predictor model that, you know, in a sense, it learns about the regulatory rules of polyadenylation in a hex cell line, but you know, also it's not, you know, I mean, because it's synthetic variation, right? It sort of escapes a lot of these evolutionary constraints. So these engineered sequences, you know, functionally, we can engineer them to do a lot of different things, but they're not necessarily gonna be very, you know, evolutionary fit evolutionary constraints. I'm not sure that's in some engineering purposes that may be required. In others, it might not. Although I do think that you can do many different approaches to sort of incorporate evolutionary constraints in order to sort of regularize these types of optimization.
Yeah, that was more of my question. Like, from a modeling perspective, have you thought about that?
Yes. So one thing that we in a later paper that I'm going to talk about, I don't have any slides for that today, but we do try to regularize optimization for a different type of task. It's more of a protein design task, where we not only maximize a fitness predictor, but we also try to maximize the likelihood of sequences given some pre-trained variational autoencoder. So a variational autoencoder can be trained to sort of capture the manifold of some sequence data set you have. For example, if you train a variational autoencoder on the native poly-A signals, you would potentially learn a model that knows about evolutionary conserved poly signals. And what's nice about this activation maximization approach is that you can place any type of model downstream of this optimization procedure, allowing you to say that, okay, we want to maximize this fitness predictor for some functional fitness. At the same time, we want the sequences to be at least this likely given this variational autoencoder. We didn't do that for any of these for the DNA and RNA regulatory tasks. We did do that in this recent paper for a protein design task, where it's really important to stay on the manifold of stably folding proteins. So did that sort of answer the question? Perfect. Great. So activation maximization, I would argue, is a really efficient method for doing direct maximization of some pre-trained fitness predictor. Another thing that's really nice about activation maximization is that it's model and parameter free, right? So once you've trained your fitness predictor, there's no more parameters that you need to tune. You can start to design sequences immediately. If you remember, there were these global scaling and offset parameters in the normalization layer, but those are trained with gradient descent as well. However, activation maximization, unfortunately, does not scale. And what I mean by that is that biologists nowadays can perform these high throughput experiments where they can measure millions of candidate sequences in parallel, of course, which means that they want a computational method that can give them a candidate sequence set design with millions of diverse sequences that are all optimized to have high predicted fitness that they can then screen in this wet lab setting. For this, activation maximization is not really a great fit, because for every sequence that you want to generate, you have to randomly initialize this nucleotide logit matrix and rerun gradient descent to come up with one sequence design. And repeating that procedure 1 million times to get 1 million sequences is computationally very inefficient. It's very computationally heavy. Also, for some of these optimization runs, you might end up in poor local optima, which means you're going to have to reinitialize and rerun the gradient descent procedure. And maybe most importantly, you're randomly initializing this nucleotide logit matrix and rerunning the procedure. That's sort of the mechanism for hopefully ending up with diverse sequences in the end. But you're running this optimization procedure independently for every sequence you design, which means that in the end, there's no guarantee that you're going to end up with a diverse set of sequences in the end. So the question is, how do we scale up activation maximization? And wouldn't it be nice if we could replace these nucleotide logits, which encode effectively just a single sequence, if we could replace that matrix with a generator, like a generative neural network that encodes an entire sequence distribution. So when you input different latent input seeds to this generator, it's going to generate many different sequences. We could then optimize the weights of the generator such that the entire sequence distribution becomes optimized for our fitness predictor. Now, if you try to do that, you're quickly going to run into the big problem of mode collapse, which is the trivial solution where the generator learns to output the same sequence no matter the input seed. Like it learns to make just one sequence. And sure, this sequence is going to be optimized for high fitness. It's going to find the local minimum in the cost landscape, but it's just going to learn to make one sequence because there's nothing that enforces the generator to be diverse here. And that brings us to this, this extension of activation maximization into generative neural networks. And so this idea that was presented last year at MLCB that we call deep exploration networks, the idea here is to jointly maximize both the predicted fitness and the diversity of the generator, essentially using Monte Carlo sampling combined with backpropagation training. So in each forward pass of backpropagation, we're going to randomly and independently sample 2 latent input seeds that we feed into the generator. The generator will independently produce 2 sequence samples from these 2 input seeds. We then take one of these two sequence samples and feed it to the fitness predictor, allowing us to optimize the weights of the generator for maximal fitness. Now, it doesn't actually matter which one of these two sequence samples we feed into the fitness predictor, because they're both generated by the same distribution, right? They're both generated by the generator. Then we take these two sequence samples and we also feed them into this differentiable similarity model that can score the similarity, or penalize rather, the similarity of these two sequences, allowing us to minimize a compound cost function to minimize both the fitness cost and some diversity cost based on all randomly sampled pairs of input seeds that we train, with standard batch gradient descent. And importantly, we start with a randomly initialized generator, and the generator model here is the only, that's a neural network, and it's the only model we train. The predictor and similarity model are pre-trained and fixed. And the similarity model can be, any kind of differentiable metric. In the paper, we try two different things. We try essentially a cosine distance between these two sequences to directly penalize, identical nucleotides. We also try latent penalties, where we use some kind of encoder model to encode these two sequences into some latent feature space, and then use a cosine distance in that latent space to penalize, latent similarity. And now in this very short video, I'm going to show you what it looks like when we train a deep exploration network to learn how to make maximally strong poly-A signals. And it's enforced to maintain a cosine distance of at least 50% between these two sequence samples. Now, on the very far left, you see this two-dimensional grid where you just see a bunch of noise. These are a fixed set of latent input seeds. So every row encodes a different input seed vector. These are a fixed set of seeds that we track throughout training, and we want to observe how the generated corresponding sequences evolve during optimization. The top graph is going to show you the predicted fitness scores for these corresponding generated sequences. On the right side, you're going to see a few handpicked example sequences generated by some of these seeds. I think the middle portion is probably the most interesting. This two-dimensional, very colorful grid is something I call a diversity plot. And essentially, the rows encode the different sequences generated by the generator that corresponds to these different input seeds. The columns denote different nucleotide positions, and the color denotes nucleotide identity. Okay, so now we're going to start optimization. As you can see in the beginning, you see these long vertical stripes in the diversity plot, which means that the sequences are not very diverse because they share the same motif across the same positions. Now, when you see as the optimization progresses, both the predicted fitness and the diversity of these sequences increases. This can be seen more clearly if we first train a deep exploration network to maximize poly-A strength while enforcing a very, the similarity loss coefficient is a very small value, meaning we don't penalize similarity very harshly. As you can see, the resulting deep exploration network does not become very diverse because you see these vertical stripes in the diversity plot. In fact, if we sample 100,000 sequences from this generator, 99.5% of those sequences are duplicates. Of course, for these few unique sequences that it does output, the predicted fitness score is pretty high. It's 400 to 1 in predicted isoform odds of selecting this poly-A signal with respect to the distal signal of our training data. However, the picture completely changes once we increase the similarity loss coefficient to a large value. Now you can see that the generator becomes much more diverse, okay? So the duplication rate drops to 0% for 100,000 sampled sequences, meaning they're all unique. And even more interestingly, I find, is that the predicted fitness scores actually skyrocket as well to a much larger range of values. So the mean predicted isoform odds now is 6,000 to 1, which is very interesting, because you would think naively that, okay, once we start enforcing high diversity in these sequences, we're going to have to trade off high fitness scores. But that's not actually the case. In fact, by enforcing diversity during training, we're promoting exploration in the fitness cost landscape, allowing the generator to probe a much larger region of the fitness landscape and learn to produce better, higher fit sequences. So exploration and fitness sort of go hand in hand here. Now, we had these deep exploration network, or DEN, generated sequences, poly-A signals, that were predicted to be, very strong. In fact, they were predicted to be 20-fold stronger than our previous, activation maximization, or AM, generated sequences. And we wanted to validate this in a wet lab setting. So what we did is we took these poly-A signals, and we performed a qPCR experiment where we synthesized the new DEN poly-A signals in direct competition with the old activation maximization signals on the same plasmid reporters, allowing us to measure selection preference between these two sequences or signals. And we measure both orientations where either the DEN signal or the AM signal is the proximal signal. This allows us to come up with an improvement odds ratio of how much preferred the new DEN generated signal is compared to the old AM signal. And so the improvement odds ratio was measured to be on average 11.6. So not 20, but still fairly high. And it's important to note that these old activation maximization generated sequences were already really strong. In fact, they were measured by us to be stronger than any of the native sequences that we assayed. And so if we extrapolate, we estimate that our new DEN generated poly-A signals are a little more than 1,000 to 1 in selection odds with respect to a native distal BGH poly-A signal that's located roughly 200 nucleotides downstream of this signal, which means it's a very strong poly-A signal. And that's actually all I had. So in conclusion, I hope I made the point that I believe that making these large synthetic MPRA data sets is a great way to induce targeted variation around regulatory mechanisms that you want to learn how that's regulated. And I kind of showed how we can learn very high quality predictive models from such data sets. And I then presented this improved gradient-based method for performing sequence design in the context of polyadenylation, and then talked about this sort of generalized activation maximizing generative neural network called deep exploration networks, and show that it can be used to produce very diverse large-scale sequence designs. And I also want to make the point that in this particular story that I presented, it was about polyadenylation, but I do think that this entire framework of going from these synthetic data sets all the way to new and improved sequence designs can be applied to many other regulatory control elements that you want to design, including protein design, which is something we're currently sort of working on. With that, I'd like to say thank you for listening. And if there are any questions, I'd be happy to take them.
So thank you for a great talk. I'll clap on the audience behalf.
Thank you.
I see there are 2 open questions. So the first question is, you note that the network uses a rich set of features, RBP motif, positions, folding, hairpins. Can you probe the learn model to learn novel insights into how APA works?
Right. That's a great question. I actually have an extra slide that I didn't show that now we actually have time to look at. So we essentially, it's a great question. And so one thing that we do in the paper that I didn't show is that one thing you can do is you can look at the convolutional filters that the network has learned. Sorry. Now the screen sharing is, there we go. So one thing to do is you can look at the convolutional filters that the predictor has learned and see what type of motifs are in there. And then, you know, you can do different types of sensitivity analysis to probe, you know, how these motifs influence selection. Another cool thing that I think we used in the paper is that we sort of took our design sequences and performed sort of additional validation experiments by doing saturation, like wet lab saturation mutagenesis on these design sequences that I think really teased out, you know, cool, important regulatory effects. The first thing that we did is we took one of these maximally strong engineered polyA signals and performed saturation mutagenesis, and the measured fold changes in isoform proportions were all, you know, negative or repressive, which is kind of indicated by the blue color in these heat maps. And I think the second validation task that we did was that really shows some of this sort of novel biology of APA. So here it's a polyA signal that we engineered to cleave at a very distant cut site with respect to the central hexamer element. And as I said, the network learned to make these long hairpins. And so then by synthesizing every possible mutation, single nucleotide variant of that sequence, we could, you know, show that this hairpin was by far the strongest determinant for cleaving at these faraway positions. And so, you know, I think it's a combination of trying to, in a sense, do different types of maximizations or probing the network to tease out these regulatory phenomena that the network has learned, and combine that with wet lab experiments such as saturation mutagenesis to validate your findings.
Okay, so the next question is, Yusuf says the results for increased diversity of solutions are really nice, and I agree. But it's still hard to judge if the PWMs that are being generated are similar in core signals, because the penalty is not, doesn't have positional bias. Do you think you can increase diversity by a different diversity scoring function that possibly accounts for positions?
It's a great question, and so kind of something we noticed is that at some point, once we increased, because what we used for penalizing similarity in this particular example is just cosine distance between the two one-hot matrices. Of course, there may be, you know, multiple alignments, or you know, if we were to take alignment into account, the actual, like the Liebenstein edit distance might be much lower, right? And you know, unfortunately, Liebenstein distance is not differentiable, but and we noticed that the edit distance kind of saturated at some point. So even though we tried to, you know, make things really diverse with a very high cosine distance, the edit distance kind of saturated. But so something that we did in the paper is that we tried this latent diversity cost, where we actually took the first few layers of the predictor up until the first dense layer of the apparent fitness predictor, and used those neuron activations as a type of latent feature space. And we then did a cosine similarity metric between these latent feature vectors in order to penalize sort of latent similarity, hoping that that's going to give us more position-independent latent features that we can say that, you know, the sequence designs have to be different for those. And I don't have any figure for that here, but we actually found that that gives us the ability to get even higher edit distances between sequences.
Okay, so there are two more questions that I'll club together. So Matthew asked, following up on his earlier question about other cell types used to test the library, do you expect your framework to apply and perform as strongly in other cell types or tissues where cis and trans regulatory mechanisms might differ? And then another question from Matthew is, do you know whether mutations affecting apparent defined poly-A signal optimality are associated with disease?
Oh, sorry, can you say that last question again?
So do you know whether mutations affecting apparent defined polyadenylation signal optimality are associated with disease?
I see. Okay, so great questions. If we start with the first question, you know, I should sort of confess that I'm not an experimental biologist. I think that, you know, that question would be better suited for the postdoc, Dr. Nick Bogard, who made these experiments. But you know, my understanding is that currently it is kind of difficult to make these large synthetic reporter assays in other cell types, or you know, in cell types that are, you know, say you know, neurons or primary cells. And you know, another thing is, you know, it's always experimentally hard to work with them because they don't, you know, divide as easily as these immortalized cell lines. I do think that one very promising approach that I do know that there are some papers out there doing that essentially combines these massively parallel reporter assays with pluripotent stem cell lines, where you can, you know, you can transfect these libraries and then, you know, push these cells to different cell states, allowing you to measure, you know, test these libraries in different cell types. Of course, this is just, you know, going to give you different types of readouts, right? Saying that this sequence in this particular cell type, like heart cells, gives you this regulation compared to neurons, which gives you this type of regulation. I think to make that even more general, I think we need to start thinking about maybe combining single cell sequencing technologies with these MPRAs in order to get a readout of both the expression levels of transacting factors in combination with the cis regulatory code of the sequences, in order to learn jointly how these, you know, transacting factors affect this poly-A signal. Hopefully that answered the first question. Now yes. So now the second question, you know, we did find in the paper, I think we mentioned a couple of very interesting variants. Actually, I do have one interesting slide, if we have time, of that I didn't get to show, which I think really highlights, you know, the it's a good selling point for neural networks and nonlinear models of regulation, because, you know, traditionally, maybe because we've had less data to work with, but you know, hexamer models where you count the occurrences of the occurrence count of hexamers in regions as a way to models to predict APA and splicing, etc. But you know, some variants, and we noted this in the paper, some variants have this kind of very nonlinear effect. Like single nucleotide variants have nonlinear effects on the regulation of polyadenylation. So for this particular example, which is, I think this is a variant in the human genome near a poly-A signal, and essentially it's a G nucleotide, that's the mutation that's introduced into the downstream region, downstream of the central hexamer. And in fact, this mutation creates a canonical CSTF binding site. And you know, any hexamer model would say that introducing a CSTF canonical binding site in the downstream region is going to increase the efficiency of this poly-A signal, right? Because, you know, good CSTF binding is very highly correlated with strong poly-A signals. So this has got to be an enhancing, like a gain of function variant. But that's not actually the case. So we sequenced, we expressed and measured this particular variant, And so the black curve shown above here are the observed measured cleavage distributions. Now, as you can see, there are actually two different cut site modes in this poly-A signal, where there's a cut site right before the CSTF binding site, where the main portion of cleavage actually occurs. And there's a weaker cut site further upstream, but that's used less frequently. Now, as you can see, which is by the observed curve here, the red curve, you see that the variant actually increases cleavage at the upstream site, but represses cleavage at this main cut site. And our hypothesis is that the CSTF binding site is too close to this cut site and actually blocks its cleavage at that site. And so the net effect of this variant is actually repressive for usage of this poly-A signal. And what's really nice, if you see the blue dashed line, that's the prediction by APARENT. So APARENT is actually able to properly score this variant effect because it's able to make use of the entire sequence regulatory code to make an informed decision of where the poly-A cut sites are, and sort of has learned that, okay, CSTF sites too close to those poly-A signals, to those cut sites, is going to repress cleavage. So yeah, hopefully that answered that question.